Welcome to today's Explainer. We're looking at one of the most fascinating and honestly frustrating mysteries in the history of modern medicine. For decades, scientists have been staring down a molecular mechanism that drives some of the deadliest diseases known to humanity. And the crazy part? They knew exactly what it was. They knew exactly how it was broken. And yet, they were completely powerless to stop it.
I want you to imagine, just for a second, that your cells are controlled by millions of tiny biological light switches. They're telling your cells exactly when to divide and when to stop. Now, what if one of those vital switches got flipped to the on position, and then the switch literally broke off, leaving it stuck on forever? Well, that simple, absolutely terrifying concept is the core mechanism behind some of the most aggressive tumors we face.
And this isn't just some rare, isolated occurrence. Alterations in the RAS family of genes, which is the family this broken switch belongs to, show up in roughly 20% of all human cancers. To put that into perspective, we're talking about an estimated 3.4 million new patients worldwide every single year whose cancer is driven by this exact malfunction. The stakes really couldn't be higher. Section 1. The vital molecular switch. Okay, let's dive into this. To understand how to actually fix the broken switch, we first need to zoom all the way down to the microscopic level and see how things are supposed to work in a perfectly healthy cell.
Meet KRAS. KRAS is the most abundantly expressed protein in a family called the small GTP-ases. You can really just think of KRAS as a binary relay switch sitting right inside your cell's membrane. It's just hanging out, constantly waiting for signals from the outside, things like growth factors, to tell the cell, hey, it's time to grow, divide, and survive.
The way this switch functions is honestly just a beautiful piece of elegant biological circuitry. A healthy KRAS protein cycles perfectly between two states. When it's bound to a molecule called GDP, the switch is completely off. But when an external signal arrives, these helper proteins called GEFs come in and swap out that GDP for a highly energized molecule called GTP. Suddenly the switch is on and the cell gets the green light to grow. But here's the absolute critical part. Once the job is done, another set of proteins called GAPs step in to hydrolyze or basically break down that GDP back into GDP. That turns the switch firmly back to the off position. It's an incredibly well-balanced cycle.
Section 2. When switches get stuck. So, what happens when a genetic mutation introduces a major glitch into this beautiful, balanced system?
In healthy cells, as we just saw, the cycle neatly alternates between off and on. But in cancerous cells, mutations occur, and they most frequently happen at very specific locations called codons 12, 13, or 61. These mutations physically alter the Kay-Ras protein in a way that those gap helper proteins we just talked about, they can't do their job anymore. The mutated Kay-Ras protein essentially loses its ability to break down GTP, and the result? The switch totally loses its ability to turn off. It is permanently locked in the active GTP-bound on state, just blasting this continuous signal that tells the cell to divide endlessly.
When we zoom back out to the macro level, the patient impact of this stuck switch is just devastating. According to our sources, Kay-Ras mutations drive up to 90% of pancreatic adenocarcinomas. 90%! In fact, research from Dana-Farber has shown that Kay-Ras mutations all by themselves are enough to initiate pancreatic cancer. We also see this same mutation in 40% of colorectal cancers and 25% of non-small cell lung cancers. These are highly aggressive diseases, often associated with a very poor prognosis.
Section 3. The undruggable protein. Now, you might be wondering, if we know exactly what the broken switch is and we know exactly how it operates, why couldn't we just build a simple drug to turn the switch off? Well, that question brings us to the real dramatic tension of this whole scientific struggle. You see, traditional small molecule drugs were kind of like keys fitting into a lock. They need deep, well-defined pockets on the surface of a protein to bind effectively. But looking at Kay-Ras, it's remarkably smooth. It's almost like a billiard ball. Plus, its affinity for GTP is incredibly high, which basically made it nearly impossible to wedge a drug in there and shut down the machinery. So this completely smooth surface led to immense frustration in the scientific community. For decades, KRAS was considered the absolute holy grail of cancer drug development. But every single attempt to target it either completely failed or it proved way too toxic for patients. It just became this widely accepted fact across the entire oncology world that KRAS mutations were simply undruggable. Section 4. Hacking the broken switch. But science rarely takes impossible for an answer.
Let's shift gears a bit and look at the massive plot twist that totally changed the history of oncology and shattered that undruggable myth forever.
The big breakthrough actually came in 2013 when Kevin Shokat's lab at UCSF made this stunning discovery. They found a tiny, previously unrecognized crevice on a very specific mutation of the protein, the KRAS G12C mutant. They called it the Switch-2 pocket, and man, this set off an absolute gold rush in pharmaceutical development. Fast forward to 2021, and the FDA approved the very first KRAS G12C inhibitors, sotorasib and adagrasib. And the field is moving so fast that by 2024, the technology had evolved even further to include degraders capable of targeting multiple forms of KRAS.
Now, what's really interesting about this slide is how these first-generation drugs actually work. The covalent inhibitors slots perfectly right into that newly discovered Switch-2 pocket. Because the G12C mutation replaces a normal amino acid with a highly reactive cysteine, scientists were actually able to design a drug that forms a permanent, irreversible covalent bond with that specific mutant cysteine. It essentially acts like molecular superglue, forcibly trapping the broken protein in the inactive off state, all while completely ignoring the healthy wild-type Kay-Ras proteins. Pretty brilliant, right?
Section 5. Cancer fights back. As amazing as that is, while these first-generation off-state inhibitors were revolutionary, we have to acknowledge a pretty harsh reality here. Tumors are highly adaptable, and cancer almost always fights back.
Basically, when you block the Kay-Ras switch, the cancer tries to rewire the whole house to bypass the blockage. Patients often develop what's called acquired resistance. Sometimes, the tumor amplifies the Kay-Ras gene itself, creating so many broken switches that the drug just gets completely overwhelmed. Other times, the tumor develops secondary mutations, things like G12D or G13D. Or it even mutates the Switch-2 pocket itself so the drug can't even fit in there anymore. Alternatively, the cancer might activate upstream receptors, literally finding biological backdoors to keep those growth signals firing.
Section 6. The future of Kay-Ras. So, how do we outsmart a rapidly mutating enemy? That brings us to the final chapter of our explainer, the next generation of therapies and exactly how scientists are responding to this resistance.
There is a brilliant paradigm shift currently happening in cancer research right now. We are moving away from occupancy-driven pharmacology and toward event-driven pharmacology. Traditional drugs, like those first-generation KRAS inhibitors, work by occupancy. They have to constantly sit in the pocket to block the protein. If the pocket changes shape, the drug fails. But this new approach is event-driven. Think about it this way. What if, instead of trying to jam the lock on a broken door, we just rip the door off its hinges entirely?
Enter PROTACs. That stands for proteolysis targeting chimeras. This is an absolutely incredible new technology. Instead of just blocking the broken K-RAS protein, PROTACs act as molecular matchmakers. They grab onto the mutant K-RAS protein on one end, and on the exact opposite end, they grab a cellular tagging enzyme. This process explicitly flags the broken K-RAS protein as cellular trash, basically instructing the cell's own natural waste disposal system to completely eliminate it.
The sheer beauty of targeted degradation is striking. The PROTAC drug links the mutated Kay-Ras to an E3 ligase complex, which then attaches a chain of ubiquitin tags right onto the cancer protein. The cell's recycling center, the proteasome, recognizes those tags and just shreds the Kay-Ras protein into harmless little pieces. And the best part? The PROTAC drug isn't even destroyed in the process. It survives and acts catalytically. That means one tiny molecule of this drug can move on to tag and destroy hundreds of broken Kay-Ras proteins. It is incredibly efficient.
So we've journeyed from a fundamental cellular mechanism straight into an impossible biomedical mystery and finally landed at the absolute cutting edge of modern pharmacology. We are literally teaching cells how to throw their own broken switches into the trash. Which leaves us with one final, hopefully inspiring thought to take away today. As our technology evolves from merely blocking these mutant proteins to destroying them entirely, what other so-called undruggable diseases might we be able to cure next?